everyone. I'm Konstancja Gałat, and I'm first-year PhD student at Laboratory of Cellular Genomics, IIMCB Warsaw. And today I will talk about my research that tries to answer the question, does the microbiome affect nutrient absorption? So a little bit about me. So I finished both bachelor and master's degree in biotechnology. So I switched to bioinformatics in some time. Actually, I found that during my master's studies, I found that I hate wet lab and I wanted to do something else. On the second year of my master thesis, master studies, I started my second master degree in bioinformatics. And the plan was I will do my second master thesis with Paweł Łabaj. I lasted 1 month. And after that, I decided to stay with Paweł and do some voluntary job, finish my master in biotechnology, and move on with my life and do bioinformatics, but without a degree. It happened. So for 6 months, I was developing snake-make pipeline. Make sure you sign it on the bingo. So I developed a snake-make pipeline for microbiome analysis. And after those 6 months, I got an offer to start an internship at Eligo Bioscience, where this time I also developed a pipeline, but this time in Nextflow. And after this, I defended my thesis, came back to Poland, and started my PhD. And also, I'm now involved in paper QC that you will hear in a second. So let's go back to science. So absorption is a process that occurs mainly in the intestine. And this is important that different nutrients are being absorbed in different parts of the intestine. And the first division is that most of the nutrients are being absorbed in the small intestine, while the water and mineral salts are being absorbed in the large intestine. And it is also important that the small intestine is usually divided into 3 parts, the duodenum, jejunum, and ileum. And again, different nutrients are being absorbed in different parts. And when you want to study the absorption, you usually need to look at the metabolite levels in the portal vein, because this is the first place where the nutrients are being absorbed. So this is the best place to look at the absorption efficiency and the metabolites levels. So my PI in her previous team asked a question whether the absorption changes in different conditions. So she was checking whether antibiotic treatment and also liver fibrosis, that I won't talk about during this presentation, affects the nutrient absorption. And as you can see, probably it does. So you can see that the levels of some metabolites are different after antibiotic treatment. Sometimes the differences are large, sometimes they are small, but still they happen. For example, the glucose levels in antibiotic-treated mice are lower, both in portal and the heart serum. So we asked ourselves a question, why is that? So there are 3 main mechanisms that drive absorption and make absorption efficient. First one is the architecture of the gut, mainly the small intestine, which is lined with villi, which increase the absorption efficiency. And also the absorption itself can occur in 2 ways. First one is paracellular transport, and the second one is transcellular. And paracellular transport involves passage of small substances between the cells. And larger substances, or with different, for example, with hydrophobic properties, they need some specific transporters that allow them to pass through the enterocytes. And in all of those 3 things can change, leading to absorption problems. So for example, in Crohn's or celiac disease, the villi are being shorter, which decrease the area of absorption. In diseases like SIBO, the distances between cells are larger, which allows too much nutrients to pass through between the cells. And there are diseases in which some transporters are being differentially expressed, which also can lead to malabsorption of specific nutrients. So we asked ourselves a question: what happens with those three things in antibiotic-treated mice? So there is a paper that tried to uncover some of it, and they found that the villi architecture doesn't change after antibiotic treatment. The paracellular transport is diminished. It is usually studied by the usage of this compound, but they did not focus on the transcellular transport. So we wanted to do that, but because doing stuff in the lab takes you more time than finding the data that is already there. So I found a cool data set where they spatially mapped, they performed spatial transcriptomics, which allowed them to map, you can see that, gene expression not only to the different parts of the intestine, but also to the different parts of the villi. But they didn't focus on nutrient absorption. This is the only plot about some of the transporters. So I decided to take advantage of that. But you have to keep in mind that they didn't use antibiotic-treated mice, but they have used germ-free mice. We will go back to that later. So first, I performed section-specific analysis. So I performed differential gene expression analysis between the different zones. And also, I was trying to see, also to do that, I performed differential gene expression analysis to see whether the expression occurs, for example, somewhere lower on the villi. That's because intuitively you can think that when the transporter is being expressed like lower on the villi axis, then its access to the nutrients also may be worse, and nobody looked at this aspect before. So I gathered all of the differentially expressed transporters, which were upregulated and downregulated and in germ-free mice. In bold, there are also the transporters that are differentially expressed on the villi axis. Here, this is a more intuitive visualization of my results. And as you can see, in the ileum, which is the most terminal part of the small intestine, the changes are the biggest, which makes sense because the lower in the intestine, the more diverse the microbiome is. And for example, this is pretty interesting that those two selected transporters, they together form a dimer. But still, their expression is changed in different parts, so that suggests that they may be doing something like also alone. They do not need to form a dimer, but we only know the aspect of their dimerization. And also, this is pretty interesting because, as you can see, there are two different transporters. One of them is upregulated and one of them is downregulated. They are from the same nucleoside transporter family, but they are working slightly differently. But to be honest, we don't know much about them, so for sure we will also try to learn something more about them. So this is the overview. But okay, the next question I had was, but what when we don't have such cool data? And usually we don't, because we don't have money. But let's use some normal single-cell transcriptomic data. And I did that. My PI in her previous team performed single-cell SPF and germ-free mouse atlas. So I used the data. I extracted enterocytes from it. I performed differential gene expression analysis. I found some interesting transporters, but I've lost the information about the spatial localization of those different transporters, which is important in the context of absorption. So the question was, is the information about the spatial distribution of cells hidden somewhere in the data? So first, I tried different tools designed for the deconvolution of different factors in the data. None of them worked as I wanted them to. So I decided to go a step back and look at the PCA results again. And I found something interesting. So you can see that different markers expressed in the different parts of the small intestine and the villi are being expressed in different parts of the 3D PCA plot. And this is it is pretty cool because when you cluster the data, for example, using UMAP, the clusters like didn't make this kind of sense. Like the amount of different information about the biological and technical variability didn't allow like to form spatially resolved clusters. But in PCA, you can see that. And you can see that when you plot this in 2D, that PC3 in this example is a good estimator of the duodenum, jejunum, ileum axis. And PC1 and PC2 together are a good estimator of tip to bottom axis. But as you can see, the relationship is not linear. So I decided to fit a curve to make it like. So then I normalized the data to have simple scores from 0 to 1. And then I was able to combine the informations together. And as you can see, those two axes allow us to say where the gene is expressed. And you can see that, for example, this transporter, SLC26A1, is expressed, first of all, the expression is lower in germ-free condition, but also the expression occurs somewhere lower on the villi, which may be important because of the contact with the nutrients in the gut. So what I have done, I have established pretty decent methodology to extract spatial information from the normal single cell data, and we will probably use it in many different things. But our nearest goals, first, we want to perform similar thing, but with antibiotic-treated mice. We want to see what are the changes in the transporter expression patterns between germ-free and antibiotic-treated mice, because antibiotic-treated mice are more medically relevant and also their immune system works. In germ-free, it is not the case. And also we want to do some research using a human enterocyte model named Krako 2. And there is pretty cool system called Transwell that you are culturing your Krako cells here, and you can treat them, for example, from the top as from the inside of the small intestine. And you can see first, you can see what passes through. You can knock out some genes, for example, in the Krako cells, and you can also the possibilities are endless, but we want to see first, we want to, for example, culture Krako 2 cells with some first metabolites, some bacteria that we find interesting, and see how that affects the absorption process. So to conclude, the greatest changes in the biogeography of the transporter expression occurs in the terminal parts of the small intestine, which is probably because the diversity of microbiome is also the largest. Single-cell RNA-seq data from the small intestine allows not only to map annotate different cell types, but also to map cells to a specific regions. And you don't need, sometimes maybe you don't need to perform spatial transcriptomics. And also PC axis can be good predictors of the small intestine's architecture. Those are people and teams I work with, and thank you for your attention.
Thank you, Konstancja. Do we have questions from the audience? I had one about the thing you skipped, that being fibrosis and the relation of all of that to the liver. Do you think that your research could highlight how the delivery of nutrients from the gut to the liver influences diseases such as liver fibrosis and non-alcoholic fatty liver disease?
Actually, yes. I'm now writing a grant for that because in liver fibrosis, patients often encounter the malnutrition, and we don't know what is causing that. And our hypothesis is that the reason of malnutrition in liver fibrosis is malabsorption. So we plan to use this approach to study the absorption, the transcellular transport in liver fibrosis and what are the changes, because this will be also important because for now patients with malnutrition are treated with some dietary interventions. But when you have like less transporters, then this may be not the best solution to treat malnutrition in those patients.
I think I know which grant you're referring to, and I want to apply there too. So we will be competing, I guess.
Oh.
But if nobody else has any questions, then thank you, and we'll take Philip's presentation.